what are your current position and field of interest? Could you please describe the group you lead, its facilities and technologies? What are the main objectives of the group, and what kind of research activities are currently ongoing? What are the perspectives for future research in the biomolecular field over the next years? And how might artificial intelligence impact research? What advances are expected, and how could they contribute to the development of new technologies and research approaches in this field? And now we move to Professor Bertrand Nadel. Bertrand, up to you.
Hello, everyone. Thank you very much to have me on board. I'm Dr. Bertrand Nadel. I'm a research director at the INSERM, the French Institute of Health and Medical Research. My lab is actually located in Marseille, at the Center of Immunology of Marseille-Lumini, and it's called Genomic Instability and Human Hemopathy. So I guess our research keywords are hematology, immunology, genetics, and now single cell. You were just talking about single cell and tumor microenvironment. I fully agree with Raoul and you that particularly through single cell, we now shift paradigm, and we know that genetics is not the only thing that can make a cell evolve towards a function, but tumor microenvironment is giving signals that constantly drive a different state of transcriptional function in those cells. So we're developing basic translational and medical research with a network of hospital and biotechs and biopharma portfolio, and this drives me to my second hat, which is that since 2018 now I'm the director of Carnot Institute. This is a French competitive label providing incentives to foster collaborative partnership between academic labs and the industry, all the way from big pharma to small biotechs, and is very, very useful. So the field of interest of my lab is the lymphoid malignancies, from decrypting basic transformation mechanism to the identification of biomarkers, innovative therapeutic targets, and of course to address unmet medical needs. We're very focused on answering medical questions. Of course, we are then very much interested to transform discoveries and not to let them in the discovery field, but as well to try to transform them into the therapeutic innovations for the benefit of the patients. And for this, we usually benefit from the Carnot Institute that I just mentioned, facilities, network, platform, business portfolio, expertise, professionalization to devise ambitious collaborative projects. Partnership with the industry, I think, is one of the very important paradigm that is going to change things in the future, because everyone has a very specific sphere of expertise, and this has to be shared. And cover the whole value chain from discovery to the market is very important. So the other very important incentive of this is that working in network is wonderful, because you have access to mostly everything you need in terms of technical facilities, technologies. For example, we have some labs in the network that are developing patient-derived xenografts not in mouse, but in chicken eggs for drug screening. And this is working much better and faster than mouse PDX, and this is very useful. So in our lab, we're not doing that, but we're doing mostly single-cell RNA technologies with many flavors. And we try to use this angle to decipher tumoral subclones, tumoral heterogeneity. And what we found is exactly what you were saying, Stefano, that genetics is not doing the whole job. Constant Darwinian evolution to subclone is not doing the whole job. Tumor cells are not static. They're not frozen in their differentiation, and the tumor microenvironment is really driving this transition from one transcriptional state to another. And we are currently trying to focus on how this functional dynamics, as we call it, is impacting the resistance to treatment. And of course, the technologies evolve, and we had to go up to the state of the art, and the artificial intelligence and the evolution of the bioinformatic technologies is taking more and more importance. And you know all the story of the blind man trying to describe an elephant by just touching the skin, the tail, or the tusk. Nobody's right, nobody's wrong. Everyone has just one piece of the puzzle. And I believe that with the development of the big data technologies, it is clear that one part of the future in the cancer field, probably in many fields, will be the ability to integrate those large data from imaging to molecular to pathology to clinical, name it. The artificial intelligence, whatever the diversity of approach it describes, I think this is a pretty vague term, but I think the machine learning and all the technical abilities that it's bringing is the power to do just that, to integrate the multiomics and to reconstruct this famous elephant from the description of its parts. I'm very confident that this will be largely contribute to patient stratification, biomarker discovery, therapeutic target identification, and actually artificial intelligence is inviting itself everywhere. It's assisting pathology, it's empowering imaging, it's inviting itself in the design of clinical trials. It's going from synthetic arms to numeric twins, digital twins. I think this has a great future. And I think right now we're in the very beginning of it, where the scientists are now trying to structure the databases and to make them interoperable. That's the first step. But it's really coming fast, and I believe that in 5 years from now will be a very different landscape. Thank you very much.
Thank you very much, Bertrand. I have a question for you. Studying peripheral T-cell lymphomas, the impression I have is that clonal hematopoiesis does represent a very important player. So in other words, there is a kind of hierarchy in the mutational landscape, and mutations which are related to clonal hematopoiesis can precede and facilitate the switch to full-blown lymphoma.
Yeah, this is true. This is something that is emerging, and we realize more and more that the actual normal mutagenesis that is occurring all the way to aging is taking, of course, part of the shaping of cancer in general, and of course hematologic cancer in particular. And this is something that, of course, we will have to study in a much more precise way, because it's blurring a little bit the landscape of what we were focused on for so many years, the genetics of the tumor. Now we have to take care as well of the genetics of the microenvironment and how it makes the whole tumor evolve. And actually we know as well that the tumor is instructing the microenvironment. The microenvironment is responding to this instruction and is shaping the tumor. So this bi-directional crosstalk is absolutely fascinating, and I think we're just in the beginning of understanding how clonal hematopoiesis and other external factors are important in the evolution of the tumor.